Hello, my name is Paulina Reyes, and this is my modeling project for Bio 201 on the propagation of action potential down the axon of a neuron. I hope you enjoy. This is a basic myelinated neuron. In teal, we have the cell body, which includes the dendrites, receivers of the cell, and the nucleus. In light green is the axon. Looking further at the axon, in pink, there are Schwann cells, or oligodendrocytes. Oligodendrocytes form myelin sheaths on neurons in the central nervous system, the brain and spinal cord. And Schwann cells form myelin sheaths on neurons in the peripheral nervous system, which includes all other nervous tissue in the body. Between these cells are the nodes of Ranvier, which play a role in propagation. In the dark green is the axon terminals, which send the messages on to the next cell. This is a closer look at the myelinated axon and the Schwann cells that are on it. These Schwann cells form myelin sheaths on the axon. Myelin is a fat and protein substance that is wrapped around the axons that is used to speed up the rate at which impulses are transmitted down the axon. The nodes of Ranvier are the gaps between the myelin sheaths where the impulses jump to. This is a resting neuron, meaning that the neuron has not been excited yet. It's a close-up view of the axon. In a resting neuron, the intracellular fluid is negatively charged, and the concentration of potassium ions is high. The outside of the cell is positively charged, and the concentration of sodium ions is high. This is a close-up view of the membrane of the axon of the neuron. The phospholipid bilayer is impenetrable by sodium and potassium ions. In the membrane are voltage-gated channels for potassium and sodium respectively. When the nerve is excited, these gates open, causing the charge of the intra- and extracellular fluids to change. This is what propagates the signal down the axon. This graph demonstrates the charges of the intracellular fluid as the nerve becomes excited, starting and ending at the resting voltage of -70 millivolts. Labeled number 1 is when the stimulus is received by the dendrites of the cell, which causes some sodium ion channels to open. If the voltage is raised from -70 millivolts to -55 millivolts, the process continues. Number 2 is depolarization. Having reached threshold, more sodium ion channels open, causing an influx of sodium ions into the cell. This influx causes the interior of the membrane to reach +30 millivolts, at which point the axon is depolarized. 3 and 4 are repolarization. The sodium channels then close, and the potassium channels remain open, which causes the axon to repolarize back to the resting potential. Number 5 is hyperpolarization. The repolarization overshoots to -90 millivolts. This is to prevent the backflow of the action potential towards the cell body when the sodium diffuses inside the axon. Number 6 is after hyperpolarization. The sodium-potassium pump brings the membrane back to -70 millivolts by pumping sodium outside of the cell. Myelin speeds up this process by letting the charges jump from node of Ranvier to node of Ranvier, therefore making transmission faster. Rather than having to go all the way down the axon, the charges can just jump from node to node, making the propagation quicker.